A novel imaging tool, fluorescent organosilica nanoparticles. This is our abstract. The new type of silica nanoparticles as a novel imaging tool, organosilica nanoparticles, has been developed by Nakamura and colleagues. The nanoparticles can be synthesized in a few of simple reactions and have a good size and dispersibility. Organosilica nanoparticles contain fluorescent dyes and are superior in fluorescent intensity to the conventional dye. These nanoparticles have SH groups on their surface, which makes it possible to conjugate them with antibodies and other protein molecules. We synthesized organosilica nanoparticles whose diameter is 100 nanometers, including fluorescein, isothiocyanate, or Rhodamine B, and then conjugate these nanoparticles with an anti-mouse IgG antibody and so on. We applied these conjugated antibodies to immunofluorescence staining to mouse brain sections. We also discussed this novel imaging tool to detect many biomolecules. The upper image is the functional hybrid organosilica FITC particles, and the lower image is the SEM image of organosilica FITC particles. Nanomedicine has been proposed as an alternative to conventional medicine in the 21st century, and its research and development is actively promoted worldwide. Organic silica particles are gaining attention as multifunctional nanoparticles because they possesses abundant useful functional groups on the external as well as internal surfaces. Organosilica nanoparticles that will be useful for imaging applications. The product features of organosilica FITC particles are following. The max wavelength of excitation is 492 nanometers and that of emission is 523 nanometers. Their surfaces are functionalized to TO group. They are superior in fluorescence intensity to the conventional FITC. The diameter of this product is 100 meters, and this product is suitable for the detection of biomolecules. The product features of organosilica Rhodamine B particles are following. The max wavelength of excitation is 560 nanometers, and that of emission is 579 nanometers. Their surfaces are functionalized to TO group. They are superior in fluorescence intensity to the conventional Rhodamine B. The diameter of this product is 100 nanometers, and this product is suitable for the detection of biomolecules. Functional hybrids of organosilica particles enable multifunctionalization with organic fluorescent dyes and biomolecules including antibodies and DNA. Here are the examples of strategies for various organosilica particles functionalized with a variety of molecules. Fluorescent probe for in vivo and in vitro use. Fluorescence labeling for cell biology. Bead assay for flow cytometry. Multiple assay. Fluorescent markers. Point of care testing, POCD, and so on. This is the production method of organosilica FITC particles. FITC is solved in dim cell with APS incubated at 37 degrees for an hour, and 250 millimolar of APS FITC is prepared. 1 milliliter of 250 millimolar APS FITC and 93 microliters of MPS are solved in 28% of 500 milliliter ammonia solution, and the bottle is shaken for 1 minute. The bottle is incubated at 40 degrees for 10 days, and organosilica FITC particles are completed. This is the production method of organosilica Rhodamine B particles. Rhodamine B is solved in water, and 20 millimolar Rhodamine B is prepared. 5 milliliter of 20 millimolar Rhodamine B and 400 microliters of MPS are solved in 28% of 500 milliliter ammonia solution, and the bottle is shaken for 1 minute. The bottle is incubated at 40 degrees for 10 days and Organosilica Rhodamine B particles are completed. Here are the methods of conjugation of Organosilica particles. At first, antibody or protein and cloud linker are mixed at this concentration and they are incubated for 60 minutes at room temperature. After the incubation, the solution gets buffer exchange step. Remove unreacted and hydrolyzed reagent using a dissolving column or dialysis methods. And then, the Organosilica particles are mixed at these concentrations with the solution and incubated for 60 minutes at room temperature. After the incubation, the solution gets washing step. Centrifuge the mixed solutions at 20,000 G 4 degrees for 10 minutes, dispose of spannant, and add 500 microliters of deionized water. The second conjugation got capping procedure. At 20 mg per milliliters, 500 microliters, total 10 mg of N2-hydroxylamine and incubate the mixed solution for 60 minutes at room temperature. Centrifuge the mixed solution at 20,000 G 4 degrees for 10 minutes, dispose of spannant and 500 microliters of deionized water. These are the examples of applications of Organosilica particles. The left figure is a fluorescence-linked immunosorbent assay ELISA of labeling Organosilica FITC with protein A. These are the conditions of this ELISA. According to this ELISA, Organosilica FITC conjugate protein A is superior to the FITC conjugate protein A in the fluorescence intensity. The right figure is ELISA of labeling Organosilica FITC with anti-rapid IgG. The conditions of this ELISA is here. And this ELISA is a comparison between Organosilica FITC conjugate anti-rapid IgG antibody and OH-capped one. According to this ELISA, the OH-capped Organosilica conjugate antibody is superior to the non-capped Organosilica conjugate one in the fluorescence intensity. This figure is ELISA example of labeling Organosilica Rhodamine B with an anti-mouse IgG. These are the conditions of this ELISA. According to this ELISA, Organosilica Rhodamine B conjugate antibody is superior to the Rhodamine B conjugate antibody in the fluorescent intensity. This figure is fluorescence maintenance rate of Organosilica Rhodamine B versus Rhodamine B. The method is the following. Prepare 1 mg per milliliter of Rhodamine B or Organosilica Rhodamine B solution in water. Drop 2 microliters of each solution on glass and evaporate their water. Measure excitation between 538 and 562 nanometer wavelengths, emission between 570 and 640 nanometer wavelengths with the excitation light irradiated continuously. According to the figure, Organosilica Rhodamine B is superior to Rhodamine B fluorescence maintenance rate. This is immunohistochemistry of Organosilica particles. In this method, paraffin embed blocks were used. Mass brain section were prepared. At the first antibody direction, anti-new rapid IgG molecular antibody was used. At the second antibody direction, two kinds of antibodies were used. Gold anti-rapid IgG uncapped Organosilica FITC conjugate or hydroxyl-capped Organosilica FITC conjugated. According to these figures, hydroxyl-capped Organosilica FITC conjugate antibody is superior to uncapped Organosilica FITC conjugated antibody in signal noise. These are summaries. We synthesize fluorescent Organosilica nanoparticles with a diameter of 100 nanometers. These nanoparticles were internally functionalized by FITC or Rhodamine B, imparting a high fluorescent intensity and fluorescent maintenance. Nanoparticles have been conjugated with an anti-mouse antibody, anti-rabbit antibody, and protein A, all of which can be chemically modified while retaining their binding abilities. The capping of Organosilica nanoparticles after antibody conjugation improved the signal-noise ratio. which may have been due to reduced nonspecific binding by the free SH group. The resultant fluorescence nanoparticle conjugate are useful as biological imaging tool. Thank you for listening to my presentation.